Lung cancer is a leading cause of cancer death. Lung cancer originates when abnormal cells grow in an uncontrolled way in the lungs. New age treatment involves targeted therapies that inhibit cancer cell growth by disrupting specific regulators that support proliferation. Such treatment regimens result in fewer side effects compared to chemotherapies. Dr. Sanjeev Das's group at NII investigated one such candidate, which shows potential for targeted cancer therapies. This is the HDAC5 protein, a histone deacetylase whose aberrant expression has been observed in multiple cancer types. In a recent study, Dr. Das's group found that HDAC5 is upregulated in lung cancer tissues, making it an effective target for the therapies. HDAC5 triggers the deacetylation of genome organizer protein SATB1, which promotes the downregulation of crucial tumor suppressor genes. The HDAC5 SATB1 axis also modulates key metabolic enzymes such as SDHA, which contributes to oncogenic epigenetic reprogramming. HDAC5 depletion inhibited the growth, invasiveness, and metastatic potential of tumor cells. These findings demonstrate that the HDAC5 SATB1 axis plays a pivotal role in modulating key oncogenic pathways and promotes the progression of lung cancer.
The paper is about studying the gene expression changes which happen during lung cancer. We have looked at one particular protein, it's a genome organizer protein, SATB1. So we showed that how SATB1 is regulated, and we showed that SATB1 can downregulate the expression of many tumor suppressor genes. So going ahead, we plan to block SATB1 function so that we can facilitate cancer treatment.